Hey Colleen, I'm having a little trouble in biology right now. Can you help me and tell me what DNA replication is?
Sure.
All I know about DNA replication is that a DNA molecule starts out looking like this, and I know that we have to get two of them. Can you walk me through it?
Sure. The enzyme helicase unwinds DNA. Bases are pulled apart. The DNA is unzipped. The enzyme primase lays down primer, which consists of RNA molecules. Elongation of the DNA strand continues by the enzyme called DNA polymerase. Another DNA polymerase goes back and replaces the primer RNA with DNA molecules. The other strand, the lagging strand, must grow in short segments called Okazaki fragments. The enzyme ligase comes along and joins the Okazaki fragments together. Replication continues on both strands until there are two identical molecules of DNA.
Thanks, Colleen. I think I'll get an A on my test now.
Yeah, no problem. I still don't get transcription and translation, though.
Well, then I'll start with transcription. Transcription is changing DNA to mRNA, and stage 1 is initiation. Located on the DNA is a sequence of bases known as the promoter. The promoter is the start transcribing signal. The enzyme RNA polymerase attaches to the promoter and untwists the DNA. RNA polymerase moves down the DNA, unwinding it. RNA nucleotides are added based on what the DNA template says. Remember, RNA has uracil, which replaces thymine. The DNA strands will form the double helix once the RNA is made. Stage 3 is termination. RNA polymerase reaches a special signal that indicates the end of the gene. RNA polymerase detaches, and the mRNA is cut free. Introns, non-coding sequences are cut out, while exons, coding sequences, are joined together in the modifying of RNA after transcription. Next, we have translation. It's the process of translating the mRNA into proteins. Proteins are chains of amino acids, and there are only 20 different amino acids. mRNA is the first ingredient for translation, and tRNA acts as a molecular interpreter, and it carries amino acids, and it matches the amino acids with codons using anticodons. Ribosomes are organelles that actually make polypeptides. They're made up of two protein subunits, and they contain ribosomal rRNA. A fully assembled ribosome holds tRNA and mRNA for use in translation. Translation. It consists of initiation, elongation, and termination. The first phase brings together the mRNA, the first amino acid with its attached tRNA, and the two subunits of the ribosome. In elongation, the first step is codon recognition. The second step is peptide bond formation, where the ribosome catalysts bond formation between amino acids. Step 3 is translocation. A tRNA leaves the P site of the ribosome. The ribosome moves down the mRNA, and this is the process of elongation.